We are going to go ahead and get started today. Welcome to Science Friday, packed house in the house, and we do have plenty of virtual joiners as well. This is always one of our most popular Science Fridays because it is the one where we're going to present, or our summer interns are going to present the projects they've been working on this summer. So we have 4 interns that will be presenting today. Eloise, Libby, Nora, and Sydney. I will let them introduce themselves, but give the background, what they've been up to. Again, for in-person folks, thank you to the Friends group for providing some refreshments. We appreciate that. And let's get it started. So Eloise is going to be first. All right, we might also get some questions from the chat, which Giovanna will let us know, and I'll read them off to you if we get them.
Sweet. Okay. Hi, everyone. I'm Eloise, and for my 10-week project, I focus on understanding Indigenous histories of Ohio.
Eloise, where did you do your research for this topic?
Where?
Yeah, what are your sources?
Right here. Giovanna was actually a really big contributor to that with resources from the library,
Okay.
and also different tribal websites and lots of other online resources. And I was in touch with the Huron Historical Society as well. They were very helpful.
All right, go ahead.
We're good? Okay, let's try again. I'm Eloise again, and I'm going to talk about understanding Indigenous histories of Ohio. So just a quick rundown of what I'll talk about today. I'll introduce myself formally, give you a little overview of what my project is and what inspired it, then talk about how I went about completing this project. I'll show you my final product, and then we'll wrap it all up with a nice conclusion. So I'm Eloise Ayotte. I'm a rising senior at Butler University. I am focusing on environmental studies, political science, and sociology in my studies. So I like to look into the intersections of social and environmental justice, which was kind of a big inspiration for this project. Science is very important, but I really like social science. So that was kind of like my goals in doing some work here at Old Woman Creek, was to let the wonderful scientists do their thing, and then I would do mine. Next year, after I graduate, I'll be getting my master's overseas in environmental justice and governance. So sticking with that theme, I just really love working with people and finding ways that we can incorporate people in our movements to preserve and protect the environment. So the people that I chose to focus on this summer were historical residents and citizens of Ohio. So when we got here, all of us interns were told that we needed to pick a topic to focus on for our 10-week period to research and then create a final product. And I had heard the legend of Old Woman Creek being that, it had roots in Indigenous history, but no one's entirely sure exactly what the true story is or where the name came from. And that kind of got me thinking, why don't we know more? Like, what resources do we have and what are we lacking in our understanding? And how can I spend my time making sure that we have more resources to move forward in the future to be able to understand these cultures and respect the past of the land that we're living and working on today? So that was kind of my inspiration for the project. And then it was just from there a lot of learning and talking and figuring things out. And so it was just lots of reading. Like I said, with my lovely question, thank you for that. Giovanna was a lovely help to me with providing resources from our library here. The issue that I was running into the most, however, was that the information was very fragmented. There would be a couple pages in a book here, maybe a chapter in a book there, a page on a website, but there was not really anything that had all of this information in one cohesive place where it was like a complete chronicle of the histories of people who lived in Ohio. So in my conversations with the people here at Old Woman Creek and the people I was working with in historical societies and who are doing this work across Ohio, it became really clear that the main issue was just that we just don't have access to a very comprehensive resource to be able to understand this part of our history. And a lot of the resources we do have are told from the perspective of the settlers and the people who still live here today, which are really important parts of the history, but to be able to really be good stewards of all the people that have lived here and all the history of our land, we have to be able to understand history from a number of different perspectives, including that of indigenous people who lived here before us. So after weeks of reading and researching and kind of just collecting information and absorbing as much as I could, I started to look into how I could put all of this information into one place in an interactive and accessible resource so that people in the future who might be interested in this topic could look into it and be able to learn a little bit easier than I had to learn, which is how I created my product, which is this interactive timeline of Ohio's indigenous histories. Let's see if I can pull this up over here. Where's the mouse? Okay. So I chronicled 110 events and different historical pieces of information into this one interactive timeline. It starts with this introduction, which I felt was a really important piece to add into this timeline to kind of introduce the ideas that would be presented here. It was important for me to give people a little rundown of who they would be learning about and when they existed, where they lived, so you can have a really accessible, easy to access list of indigenous groups who have lived here in the past and are still present today, as well as some significant historical individuals, figures, or groups that will appear in the timeline. Another thing that I chose to include was a language acknowledgement. One thing that is very important to understand when dealing with understanding history and honoring culture is that we haven't always been as respectful as we are today, but that doesn't mean that we should ignore that part of history, because to get a complete and accurate representation of history, we have to come face to face with the times that were maybe a little bit harder to learn about. So this is just to acknowledge the fact that in the past, some terms have been used that are now considered harmful or outdated, and while I do not agree with the use of those terms, today, it is still important to recognize the history behind them to be able to move forward in a more respectful and culturally sensitive way. And then I included a content warning as well. I felt that was important just in the context of things like wars and massacres that, again, are really important to understanding the history of how we got to where we are today, but can be a bit upsetting to some audiences. So my timeline goes from 700 BCE with the Adena culture, which is one of the mound building cultures who lived here in Ohio. One of the earliest cultures we have proof of here in Ohio. There are mounds that still exist today. And then it spans all the way to 2025 today. So it starts with these prehistoric cultures, and then you can manipulate the timeline as much as you want. Being 110 events, I'm not going to run through every single thing with you today in these like 7 minutes, but I will go ahead and highlight just some important things. So some of you may be familiar with the Beaver Wars that started in 1628. It was important to me to chronicle all of Ohio's history with just a focus on the Lake Erie region, since that's where we're all living and existing today. So the Beaver Wars, for example, is one of those events that impacted the entire state and the entire nation, but was a lot more central to the Great Lakes region since it had a lot to do with the trade routes that were accessible in the lake. And then we kind of move forward. You can see the events that stretch longer are ones that just span longer periods of time. And then there are other events that just happen over 1 year. Some are more significant than others, so they're a bit more context heavy. For example, Ogons was a chief here of an Ottawa village. He was very popular in the Sandusky region, so his information, he comes up a couple times in the timeline. He's highlighted in that introduction, like I pointed out, so just kind of trying to focus on our region to have a better understanding of like what happened in our backyards, but also recognizing that lots of different events had significant impacts. So let's see. Here's Old Woman Creek, for example, with the legend. I was curious about it. It's still, not entirely clear what happened. There are really 2 legends that kind of float around. We may never be able to fully prove what it is, but it's still really interesting to look into and understand that history. So I did include that in the timeline for people who are curious. We can see our historical markers are significant in these things. You kind of move on, and then you get to these more complicated issues with removals. The Indian Removal Act was passed in 1830, and then for about 15 years after that, Indigenous people were moved out of Ohio, which is why we don't really see a huge presence today. But that's another really important thing to understand in the timeline is when that happened and why it happened. And then from there, what happened in the 100 years that followed, 150 years that followed to kind of help repair that damage that was done. And that's where we get into more of the like national context with things like the Indian Citizenship Act. We have the Indian Reorganization Act. There's lots of different, this is kind of where government influence started to step in and try to work to recognize more sovereignty of these nations and honor the damage that had been done and worked with reparations to kind of move forward. We see things like Indigenous Peoples Day, which started in 1977. We still honor that today. So just kind of things that impact mostly Ohio, but also on a national scale that have really important context in understanding our history and our future of honoring these cultures. And then at the end of my timeline, you'll find just a little conclusion, kind of wrapping up the timeline and urging people to understand that this is a really important thing to look into to learn about our history, but it's also important when we're looking to our future to be good stewards of our land and citizens of each other and good to the people that we live with and the land that we live on, and recognize we don't necessarily have ownership of this space. We have not always had ownership of this space, and it's important to recognize that, to work to respect and elevate cultures as we move forward in our work to elevate the land. And then I also included hyperlinked sources in the timeline, so if anyone ever wanted to go in and manipulate the timeline and learn a little bit more, there are lots of sources that I found really helpful. And so that's just kind of the product. Let's see if I can get this back. Yeah, so looking forward, it's not currently publicly accessible because it has to go through some edits and things like that, but it is hopefully someday going to be a resource that Old Woman Creek puts out to the public. But even if it's not, I do heavily encourage you all to take some investment in the place that you live in and the history of the place you live in, and recognize that things have happened in the past that maybe aren't always super awesome to learn about, but it's really important as we move forward and work to be really good citizens of this land, especially if we're going to try to be stewards of this environment that was not originally ours. And yeah, I just think I'm really grateful for the time I got to learn with this stuff, and it's definitely a working, living project. So I hope that in the future it will grow and expand so that people can learn a little bit more. And I would also really just like to take a second to thank everyone who helped me out along this process. All of the staff at Old Woman Creek was incredibly helpful in getting resources with me and bouncing ideas off of. I'd like to thank the Friends of Old Woman Creek. They're the reason I'm here this summer, so thank you all so much for bringing me here. And yeah, it was a really great 10 weeks, and I'm super excited to have this product, and sad that it's over. So yeah, thank you. Yes.
So it's not going to be on, well, maybe this question is more for Jen than for you. It's not going to be on the website right away.
Correct.
Yeah.
Is there a way that we can get this emailed to us or something if we were interested in reading it?
Sure, it's been fully vetted and judged by department communications staff. So yes, in the future.
Okay, thank you.
Yeah. Yes.
Is this session being recorded, and can we access this session sooner than that process won't happen?
It is being recorded. We'll have to see what the final product looks like. But you can probably get a private recording from me if it doesn't get fully published.
Thank you.
Yes.
Did you run across any references to the site being called Old Woman's Creek?
Yeah, that was part of, I believe, the first legend. It was like the initial legend was like Old Woman's Creek, and then it had evolved just like through oral storytelling. It like got convoluted in the histories of like people relaying the information and things like lost in translation, and that's when it started to be more prominently like Old Woman Creek.
Another question. Did
Yeah.
you find anything out about the skull that was found over by the mouth of the creek?
Yeah, that's why people tend to lean towards the second legend, because they like to think that that's like the most scientific evidence that we have linking it to the second legend, which you all can access. That is actually already on the Friends of Old Woman Creek website. So the second legend is the one where a woman passed away at the mouth of the creek, and that's how it got its name. And then they did find a skull that is believed to have belonged to an indigenous woman, and that's why people tend to, in my experience in research, lean towards that second legend because they think that that is like the proof that the second legend is more accurate.
Skull.
I did not.
It's with the tribes. So Ohio History Connection, which is the history group for Ohio, had it and made sure it made it back to the indigenous people, so she could be with them.
Exactly where it is, but the indigenous people have it somewhere.
Yes.
Thank you. Yeah.
I was always told when I was young that Route 2 got put in later or was held up because of indigenous remains were found during the scope of when they were trying to,
I found like around that time period just people who were like involved in digs looking for things like that, so I could see how that would like line up and have caused like a delay in that, but I didn't specifically look into anything with Route 2's construction.
I always remember that.
I think that would make sense just from like other resources. The timing would make sense, but yeah, it wasn't like necessarily part of the project looking at that Route 2 project. Yeah.
Great, thank you, Eloise.
Yeah, thank you.
Needs to have that red line around it. Research that, yes.
Wait. Excuse me, sorry. Just trying to find my cursor.
It's on the screen. And you can use the arrow on the keyboard.
Okay. So, hello. Hello, my name is Libby Travis, and today I'm going to be talking to you about interpreting research at Old Woman Creek. So first off, a little bit about me. I am a rising sophomore at Bryn Mawr College, and I'm studying biology and environmental studies. This summer, I spent as the education and interpretation intern here at Old Woman Creek, and so you might have seen me at the front desk or on canoe tours or helping with other programs. So a bit of an overview about Old Woman Creek. Old Woman Creek National Estuarine Research Reserve's operations are broken down into 4 sectors: education, stewardship, training, and research. And this summer I chose to focus on research because there's so much that falls under the research umbrella that the public just has no idea about. And so I really wanted them to start thinking about it and start asking questions about that side of Old Woman Creek. So a few of the problems I found with the public knowing about research or their lack of knowledge about research was, firstly, scientific research is hard to understand and it can be difficult to interpret for people. That is, explain it to them and also show them how it can relate back to their lives. Additionally, right now with our resources, we're only reaching auditory learners because most of what we tell people is through word of mouth on canoe tours or in the visitor center. And so that can be challenging for people who aren't auditory learners. Third, there are limited opportunities to tell the public about research at Old Woman Creek, just because it's on like canoe tours where we only have 20 people and a lot of them are repeats, or on Science Fridays, which is not always talking specifically about Old Woman Creek, and there's not a lot in the visitor center to prompt people to ask questions so we can have individual conversations. So a solution I was looking for, and I think I found it in passive interpretive materials. These are materials that
require no interpreter input to communicate information to the public. That is, they can exist and we don't need a naturalist or education staff to help people understand this information. The materials can do it themselves. And that can take the form of brochures in the visitor center, like Backyards for Butterflies, or the H2 Ohio Working Wetlands brochure that we have hanging up on the walls. This can also take the form of the displays in the visitor center as you walk around that are helping you understand Old Moon Creek and how it relates to your life. And so I wanted to create a passive interpretive material that people could take with them, so it would be more similar to these brochures that I am displaying. And why do I think that passive interpretive materials are the solution for this situation? First, they ease the interpreter load, especially if we have large groups coming into the visitor center that are maybe separate or not all together. They can get scientific information about Old Moon Creek without us having to have a one-on-one conversation with each person in the visitor center. So they have the opportunity to learn even without one-on-one support from interpreter staff. Secondly, they prompt curiosity. I find that a lot of times people need a little bit of a foundation of knowledge to be able to ask questions and figure out where they want their inquiry to go. And so with these materials, they could read a little bit about the research and then form questions and ask us, and then we can have an individualized conversation that really helps them with their journey of curiosity. Thirdly, they're clear and concise, and so by putting in the work to make something clear and concise about research, it can then be better understood by people and also help with what I was talking about before, prompting curiosity and good question asking. Finally, they are long-lasting. Specifically with the material I wanted to create, I wanted people to be able to take it home with them so that they could remember what was there and they could use it as a memory aid, but also so that they would be empowered to tell other people about the research here. And so they have that source where if they're unsure about it, they can still feel confident talking to other people and engaging in those conversations outside of Old Moon Creek. So I selected 3 research projects to highlight on this material, and those are the Compass FME project, the Exchange project, and the Rewriting the Redox Paradigm project. And I chose these because, first of all, they are large projects. They are Old Moon Creek in collaboration with other institutions like Kent State, and specifically Compass FME and Exchange projects are through many institutions across the United States. And so they have a really big impact on our understanding of wetlands on a national level. Secondly, they are visible to the public. All 3 of these projects do sampling in the cove that is along the blue and red trails. And so I wanted people to be able to have that connection. Like if they're walking along the trails and they see like a tree that has rope around it, they can be like, oh, that's part of this project. Or I know a little bit about why that tree has a rope around it. And third, they introduce new terms and concepts. And I really wanted people to be enabled to continue to interact with this research and to continue to explore it on their own by looking it up online or stuff like that. And so I chose to highlight 2 main concepts for vocabulary to help people with that. The first is the terrestrial aquatic interface, and so this is an area of transition between land and water, and Old Moon Creek is an example of that. And while that might seem like a simple definition, the terrestrial aquatic interface is anything but that. And so both the Compass FME project and the Exchange project are looking at that terrestrial aquatic interface and how it responds to different changes, but I'll get more into that later. Secondly, I'm going to be talking about redox reactions, which are chemical reactions that control wetland processes, and these happen throughout the wetland, and they control stuff like greenhouse gas emission and nutrient retention, and they are basically the transfer of electrons between different substances, and so they seem like invisible, like we can't see them happening, but just like electricity, they are happening and they are a big factor in wetland processes. So the first project I highlighted was the COMPASS FME project, and this stands for Coastal Observations, Mechanisms, and Predictions Across Systems, Field Measurements, and Experiments, which is a very long title. But this project is considering how flooding affects interactions between plants, soil, and water along coasts, and they can collect data on a variety of things like sap flow in trees, natural gas flux, and meteorology, the weather, the wind, stuff like that, that impacts how the wetland functions and to understand how it responds to different events. And so these are the sampling that you might see when you're walking along the trails. If you see a tree that has something on it, like the collar on the bottom that's measuring gas flux, or if it has like a rope around it at the base, then that tree is probably being used for this project. And on the left, you can see how the main focus of this project is because our current understanding of the terrestrial aquatic interface and coastal systems is very simplified because we have a strong foundation on land and we have a strong foundation on water, but the mixing of it is what we're really trying to understand and get at. So the second project is Exchange, and this stands for Exploration of Coastal Hydrobiogeochemistry Across a Network of Gradients and Experiments. Once again, another very long title. Exchange is a sub-project of COMPASS FME, and so they are focused on a variety of sites. So COMPASS FME uses multiple sites, including Old Woman Creek, both in the Great Lakes region and the Chesapeake Bay region, and Exchange expands on this. And so they have a lot more sites, 52, I believe, and they're able to do this by collaborating with researchers around those areas. Like they will send a soil sampling kit like the one seen here, and they will send it to Old Woman Creek, and then Old Woman Creek research staff will collect the samples and send it back. And Exchange is really focused on open science, which is trying to make science more accessible and collaborative, and they do this by publishing their data sets and encouraging collaboration across institutions. Last but certainly not least, rewriting the redox paradigm, dynamic hydrology shapes nutrient and element transformations in a Great Lakes coastal estuary. So this is tying back to the redox reactions that I was talking about earlier. And right now, when we use redox, our understanding of redox reactions to estimate wetland processes like greenhouse gas emissions, we use a very simplified wetland model, which you can see on the left, and that doesn't account for any changes in water level or changes to the soil that might be because of plants or other things that are in the soil. And so this project is focusing specifically on a very small scale, collecting data on how redox conditions are variable throughout the soil, and then using what they find out through that to scale up and model entire wetland redox processes and see if that makes a difference in our understanding or estimations of processes like greenhouse gas emissions. So what did I learn through this experience? First, interpreting research is really difficult. It took me a lot of time to kind of collect all my resources, orient myself, try to understand these research projects first before I could even think about telling them to other people, which thank you to everyone who helped me with that. Secondly, there's so much research happening here on the reserve, both specific to Old Woman Creek and in with collaboration with other institutions. And so I'm just scraping the surface. I'm pretty sure there are like 49 research projects that have started since 2020, which is quite a lot. And third, interpreting research is really valuable. That connection of understanding what's going on behind the scenes and that deeper understanding of Old Woman Creek, I found that this really helped me connect to Old Woman Creek itself, but also give me the tools to orient myself and engage both with the outside world around me, but also be a more informed decision maker and make my own decisions with the support of the information that I learned. And so I really hope that when this material does get cleared through the DNR branding and such, it will help people ask more questions, engage in more conversations, and be more curious. So that's what I would like you to take away from this, is that like curiosity is really driving this. I wanted everyone to have the tools that they needed to continue to be curious and to point their curiosity in new directions. And I think that this interpretive material will really help with that. So a big thank you to everyone who has helped me this summer and supported me both on this project and in general. And a really big thank you to Friends of Old Woman Creek for giving me this opportunity and allowing me to be here for the summer. And here's my bibliography, and does anyone have any questions?
You did a good job with tongue twisting titles.
Thank you. I practiced a lot.
It is really hard to understand research, and if people, need to make decisions, informed decisions and stuff, and it's not gonna help if they don't understand anything about it.
Exactly.
And when people who come here understand more of what's going on, that, I don't know, just more informed. Thank you. It was a hard topic.
Thank you, Libby.
Yes, thank you, everyone.
Oh,
Oh my god, can I have it? Whoa. How do I get it?
Escape.
Escape.
Escape. Escape.
Oh, perfect. You need it. Oh, okay, that's fine. Okay, we'll figure it out. Okay. Hello, everyone. My name is Nora Haddon, and today I'm going to be discussing the research that I had the opportunity to conduct while I was here at Old Woman Creek. My research has been focused on submerged aquatic vegetation and completing a survey. So a little bit more about me. I am a student at Kent State University. I'm going into my senior year there. I have been involved in research for about 2 years now with the Kinsman Costello Lab, studying biogeochemistry. And this year I have spent my summer here at Old Woman Creek as the research and monitoring intern, and I have been able to complete my own project here. So to begin, what are wetlands? Just a bit of background here. So wetlands provide a lot of important ecosystem services like nutrient retention and cycling and carbon storage. They are defined by 3 factors, one being filled with water, hydric soils, having water for most of the year, having organic soils, and having specific vegetation that is there. Additionally, they are really important to productivity and biodiversity. There are a lot of different species of animals and plants that are present in wetlands, and I'm going to be focusing a lot on plants. So Old Woman Creek is a wetland ecosystem on the coast of Lake Erie, as many of you very well know. It is controlled by a barrier beach, which is a very unique aspect to this system, and meaning there's a lot of variation in water level. That barrier beach opens and closes a lot during a season, a lot more during a year. So it's impacting what's going on. So there are also very turbid waters here, making it hard for certain species to live here, also impacting temperature, nutrient retention, and a lot of other aspects of the wetland. But here we have a lot of vegetation. If you go out there right now, you're going to see all the blooming lotus. They're taking over everything. We have 3 different categories of vegetation here. We have emergent plants, which are the ones out of the water, like our Phragmites, our cattail or bulrush. Then we have our floating vegetation, like our water lilies and American lotus, and our duckweed and things like that.
And then we have our submerged vegetation, which I will discuss a little bit more in a second. So here at Old Woman Creek, we do vegetation surveys every few years. As you can see here, I have located where these transects are. We have one up here, one down by the middle, and one back by the railroad tracks. These are done every few years, and they focus mostly on those emergent and floating species. We don't focus as much on the submerged species. There has been previous research done here on submerged species, but it hasn't been done in quite a few years now, and with changing environments and water levels, it could impact what we've seen in the past. And so I kind of wanted to take a deeper look. So what is SAV, or submerged aquatic vegetation? They are plants that grow below the water's surface. They're kind of hard to see, but they're great habitat and food for animals in the estuary. These are things like our pondweeds. So down here we have like leafy pondweed or thinner pondweeds that you may think of. This is a type of pondweed, but they look vastly different. And then down here we have a plant called coontail. They grow in coastal areas a lot of the time, and there are both native and invasive populations. One notable invasive population is hydrilla. It is being found more and more commonly in Ohio, and in these wetland systems, it takes over in really dense patches and chokes out all the native species that are really important to biodiversity and sustaining the ecosystem's nutrient cycling and overall practices. And so it's really important to understand like how many invasive species there are, how many native species there are, and what we can do to help. So that kind of brings me to my project. I wanted to understand, since we have a lot of other monitoring going on, we look at a lot of those emergent species that are invasive, like our Phragmites. We monitor and maintain those populations as needed. And we also do like frogbit polls for our floating species, but we don't have anything in place for our submerged. And I was just wondering if there were a lot out there and if it was something we needed to look into to take more action. So my first question is, how does prevalence of non-native or invasive species to native submerged aquatic vegetation compare in Old Woman Creek? And my second question was, how does richness of native and invasive species change across Old Woman Creek? So richness is just how many species are found in a specific area, and I will get into that a little bit more. So to conduct my study, I did a survey of the estuary. I set up 3 transects, which were each 100 meters long and had 10 points on each. I used a quadrat system. So down here, you can see an example of one of my quadrats. And there's a map here of all my transects. So the first one was down here by the railroad, second up here, kind of where we have our vegetation monitoring already, and our third over here in this cove off of the boat ramp. So from these quadrats, I did a standardized collection. I tried to keep it as consistent as possible. I did a raking system. You can see me here raking up some submerged aquatic there off the side of the boat, and I would collect it and then take it back to the lab. I would weigh it out to get a wet weight. I would identify it by the flowers or the plants that I can see, and then I would dry it and get a biomass for all of them. So my expectations for this project was that there was going to be a lot of diversity. I did an initial scoping of the estuary and I saw about 5 species, and I was really excited. I was like, okay, I'm going to find a lot more, because that was just an initial search. And then I expected easy collection and identification, which was wishful thinking. So to get into my results, what did I find? I am answering my first question here about prevalence of invasive to native species. I took the biomass readings and I put them all together across all 3 of my transects. I found an average biomass of invasive and native species. And here you can see the native species significantly outweigh the invasive species, but there's a lot of variability in my results. So I kind of wanted to break it down a little bit further. Now this graph kind of answers my second question, but I'm going to break it down a little bit first. So I broke it down further by transect, and then I found each species in each transect. As you can see, I only found a total of 3 species. I had coontail and bladderwort, which are both native species, at transects 1 and 2. And coontail is significantly outweighing those other ones. It is a lot more found, especially at transect 2. And then at transect 3, I did in fact find a invasive species, which is curlyleaf pondweed. But as you can see here, it is still not quite as high as that native coontail species. So there's still less of it than the native, which is very good. So to get into the richness a little bit more, I broke down at transect 3 by point. So I have marked here in orange the species that are invasive. As you can see, at this point, I found a total of 3 species, but only 2 species at any given point at maximum. And at some points, I didn't find anything. This is not bad because it's not like curlyleaf is at every single point and it's not taking over, which is really important to note. So, conclusions. Most of what I found was a native species, which is really good. That means our invasives aren't taking over, they're not choking out what we have, and they're letting diversity happen there. My study was conducted from May to July, and so that just indicates that during that time I didn't find a lot. We found that coontail was the dominant species there, and that's really important to note. It's possible that I didn't find other species because they are a more target food for things like carp that are living in our estuary, or they just weren't found in the transects that I chose. Only had 3 transects. If I had more, it would have given us a bigger scope of what's actually there, and that would be a little bit more insightful. But this project kind of sets up an idea of what we could do for future monitoring or research here, pushing like a standard or a start at least for looking for submerged aquatic vegetation. And I also noted that it didn't really seem necessary to take action with what I found being invasive because although it was there, it wasn't there in mass quantities. Some challenges I faced with my research. There wasn't a ton of recent research. One of the more recent boots on the ground studies that was here was in 2009, and the terrain and ecosystem has changed a lot since then. The water levels are completely different, and so that could have impacted what's able to grow and what is prevalent here now. Additionally, I had difficulty identifying plants. This was my first time working with submerged aquatic vegetation, and there's a lot of minute differences between species, so I had to learn a lot in just these 10 weeks to be able to look at them and kind of figure out what they were. I had a lot of books, a lot of help. The internet is a great resource. But also, some plants weren't really easy to see. Here I have a picture of one of my quadrats where you're looking down and it's all lotus. I can't see anything below it, but there were a lot of cases where there was stuff growing beneath it. So I just didn't see it. And then additionally, this was our view for most of our transects. Really, really dense, tall lotus. And the water level has been super low all summer because the beach has been open. And so we were like dragging ourselves through tall lotus trying to get to all my points. So in the future, if this research was to continue, I would love to do more studies on and surveys during times where the beach is open versus closed, or an annual survey to see if there is changes over different years and over time. As I mentioned, the ecosystem has changed a lot in just a few short years, so not knowing exactly what may happen in the future, that would be really important and could be very useful to understanding what's happening, nutrient cycling, all of that. Additionally, a seasonal survey. Some of these plants that I kind of saw at the beginning of the summer, I didn't see as much of. Maybe they were seasonal plants. They just were out of their growing season and weren't as prevalent once I actually surveyed. So going out multiple times to the same transect throughout the season might be beneficial to see if there's any seasonality differences between when these plants are growing. And then just adding more transects to get more points, because the bigger your data set, the more you're going to understand. So only having 3 transects and a total of 30 points to look at, that's a lot, but there's so much more I could have done if I had more time over the summer, because time is in fact a factor here. But I'd like to give a special thank you to all of the people in the lab who have helped me and set up my research and figure out what I'm doing. And a big special thank you to Sydney and Eloise for helping me with my sampling. It could not have been done without you guys. And then these are my sources, and thank you so much for listening. Questions?
Well, I just
Yeah.
had a comment. I'm glad to hear that that you did, if you did find early leaf, we've never seen that.
No.
So be more aware of it, because we do the muskrat monitoring.
Yeah.
And we do actually do the plants
Mm-hmm.
with the muskrats, and we do the squares and stuff. But it all depends too, because some of the vegetation disappears as the huts are built, because they end up on the
Yeah.
huts of the lodges. Very interesting, thank you.
Thank you.
Thank you, Nora.
There we go. Thanks. Okay, hello everyone. I'm Sydney Ensley, and today's my last day as a research and monitoring intern here at Old Woman Creek. And my project this summer has been focused on carbon storage. So about me a little bit, I'm a student at Ohio State and I'm studying environmental science and hydrogeology. I pretty much just love rocks and soil and being outside, so I chose my project based on how I could best suit those interests. A little background on my project. To understand soil organic carbon, it's important to understand the carbon cycle a little bit. And this is going to be the whole cycle. It's just the parts that I think are important and the parts I'm going to refer to. So in living plants, they'll absorb some CO2 from the atmosphere and store it in their biomass. And they'll secrete carbon through their roots. So that's one way that carbon gets into the soil. And then dead plants and other decomposing organisms and their waste also contribute carbon to the soil. And as you can see, this soil, I tried to make it look like it was covered in water because I want to refer to this in a wetland setting. And the saturated soil helps prevent the carbon from coming back out of the soil. So why is soil organic carbon important in the first place? It improves soil stability, helps to retain water and nutrients, and improves microbial activity, and just as a whole helps to buffer against any environmental changes. But the most important aspect, and the aspect I'm going to focus on today, is how soil organic carbon acts in terms of carbon storage, because I think it's important to look at this exchange between the atmosphere and soil when we talk about climate change and trying to control the amount of greenhouse gases that are in the atmosphere, because CO2 is a greenhouse gas that's contributing to climate change. So I did my project here at Old Woman Creek, which is such an interesting place to do soil sampling because it's really variable. It has both lake and stream input, which means things like water level and flow change all the time. And things like nutrient loading, because we're a predominantly agricultural watershed and we have a lot of storm events, as you can tell recently. Nutrients are constantly being put into the creek, as well as vegetation patterns change seasonally and even annually. So all of these factors go into storing carbon, and finding out how they do is something that I thought was really interesting. So my research question was, how does soil organic carbon vary with depth and across site conditions at Old Woman Creek? So initially I thought that the points that were furthest from the main channel would store the most organic carbon, and that's just because when I did an initial survey, I saw that most of the vegetation was further from the channel, and I was thinking, going back to that carbon cycle, more vegetation input, more organic carbon. And then I thought that organic carbon concentrations would be the most at the surface, or they would decrease with depth, because again, when a plant dies, it's just at the surface first. So that's what I was thinking with that. And so onto my methods. How did I do this? For site selection, I actually used Mike Back's sites. He's our Margaret Davidson Fellow, and he's doing a project on sediment nutrient dynamics. And he chose these sites because he's interested in how they change in terms of the inflow and outflow of the estuary. So you can see that like this yellow arrow is the inflow, this is the outflow, this is the dominant flow path or the main channel. And then these red arrows are the transects. They're labeled by number, and then there's points at the transects that indicate sampling sites. So A is closest to the channel and D is furthest away. There's 4 transects, and I did 4 points at each one, so that's 16 soil cores that I took. And you'll see I had a little mock-up of a soil core here to indicate I was going to sample to 50 centimeters, so 5 segments per core, so 80 samples total. But I ended up only getting about 68 just because some of the soil is really loose and it wouldn't stay in the core. But I got a maximum of 50 centimeters at most sites. And here's just some pictures from the sampling. As you can tell, it was super densely vegetated, and the water level, like Nora mentioned, was really low most of the time, so couldn't get the boat out there. I just had to walk. And you can even see in this picture here, you can't even see me. The lotus are so high. And just here is a basic overview of how I process the samples. The processing started in the boat. So these first few pictures are actually in a canoe. We would start by taking the soil out of the core and separating it into those sections that I indicated before. We would mix up the soil and keep it in these vials. Then we would transfer it back to the lab and dry it, pulverize it so all the particles are about the same size, sift it even further, and then I would react it with acid to remove any inorganic carbon. Put them in the carbon analyzer, which worked by combusting the carbon at about 900 degrees Celsius or 1600 degrees Fahrenheit. So very, very hot. And then just to get into some results, I do want to preface this by saying my results were run through a statistical analysis, and there wasn't any significance or statistical significance in them, but there are still things I can say about the average trends that I see. So by depth, which was my first question, it appears that the average concentration of carbon decreases with depth and then pretty sharply increases again at about 40 to 50 centimeters. But again, these standard deviation bars are really large and they overlap. So that just indicates really high variability in between my samples. And I can't really say that these points are different from one another. And it's kind of the same story with the by distance from the channel. So these A, B, C, D points. In the average trend, it appears that the SOC increases as we get further from the channel, but these error bars are pretty large and they overlap. So I can't say with confidence that these points are different from one another. So I decided, just because I was curious, to see if the soil organic carbon changed by transect, and it did a lot. So transect 1, which is up at the top here, had more than double the amount of organic carbon stored than the other ones. So I was just curious as to why that would be. So I looked into the wetland classifications of the estuary, and these first 3 are where our transects are. So you can see that this purple one is transect 1. That's the one that stored the most. And I think that the key factors in why it stored the most organic carbon was that it's a palustrine and permanently flooded site. So palustrine just means densely vegetated. Most of those photos I showed from before, where I was hiding in the lotus, those were from transect 1. And then obviously permanently flooded, it's covered with water all the time. And so I think that the fact that it was consistently covered with water, whereas the other sites might not have been, and that there was more of that vegetation input than there were at the other sites, was the main factors in why they stored so much more carbon. And although the differences that I observed were not statistically significant, the trends that I observed are consistent with other studies that have been done in the estuary. So for example, this is a study from 2023, and they also observed that at the shallowest level and at the deeper level in a soil core, that stores more carbon than the intermediate level. So kind of like the graph I showed before, and both with the location within the estuary. So they observed that further away from the main channel, so in this outflow and backflow, that has more organic carbon than this mid-estuary location, which for their study was near the main channel. And this just shows that hydrologic conditions probably influence how soil organic carbon is stored, but in terms of my study, the high site variability or just my limited amount of samples could have prevented my data from being significant. Also, just in general, the estuary has a lot of sediment turnover from the changing water level and flow, as well as storm inputs. So carbon being stored in layers can get mixed up all the time, and trends can get obscured. So that could be another reason for why my data was really variable. And then some challenges that I had, other than the sampling being really physically strenuous and dirty, which I don't mind, the fieldwork was it took a while to get the hang of being efficient with it. Our first time going out, it took like 5 or 6 hours, which is not ideal, but we figured it out. And for just as an example, this is my core at the very beginning. This is the first transect, and this was it by the end. We lost some pieces because it worked so well at keeping the soil in the core that I couldn't get it out. So I had to modify it. And so that was like field challenges, but just the main lab limitation was I kind of ran out of time. The processing takes about a week for one transect, and I had 4, so that's 4 weeks, and I had a 10-week stint here. So if I had more time, I would have done more sampling, but I didn't. And then if I were to continue this project, I would use the same transects and look at how it changes seasonally, because vegetation and water level and storm frequency, all of those change seasonally as well. And I think doing this project for a longer period of time and just getting more cores would help indicate how disturbances happen, and we could eliminate some of that error that we saw with my data. And then just a big thank you to all the lab people, everyone that helped me with my project, especially Nora and Eloise, because that field work was hard. And then does anyone have any questions? Thank you.
Sydney, we did have a question from the chat.
Mhm.
Can you please define inorganic versus organic carbon for them? And can you remind them which acid you use to get it all?
Yeah. So organic carbon is the carbon that's going to come from the living matter. So like plants, any other organisms, those are those carbon chains that make up all of us. Inorganic carbon are like carbonates, so like salts, things that are like mineralized, things that are in rocks. And I used hydrochloric acid to get rid of the inorganic carbon in my samples.
Any questions from the room?
Yes?
You and Nora both spoke about if you could do more seasonal study, which might be beneficial.
Mhm.
I'm wondering if the extremely over typical temperatures that you experienced during your studies, of course, seasonally there would be, but the extent of the temperatures, do you feel that could have had any impact on? You don't both have to answer that right now, but I'm just throwing that out. Is that a possibility?
Definitely. I think that temperature definitely had an impact, especially with carbon, just because some of the sites had really low water levels already, and the higher temperature increases how much carbon is getting broken down. So more carbon is getting released from the soil when the temperature is higher versus when it's not. So if it were cooler, I might have observed more carbon in the soil. So yeah, temperature definitely affects this aspect.
And the follow-up is that the influence of the lake, and that's why you may have seen some higher carbon right at the mouth more. Would that make sense?
Yeah.
If it was because it's cooler,
Mhm.
potentially because it's by the lake.
Potentially, yes.
Okay.
I do think a bigger factor was the higher water depth, but for sure that could have also been a factor.
All right, thank you very much, everyone. All right, again, thank you to the Friends for refreshment and also for supporting two of the interns. So Eloise and Libby were supported by our Friends group. Again, they wouldn't be here without them. September 5th is our next Science Friday, and it will be on the topic of a potential new National Estuarine Research Reserve in Lake Michigan, the Green Bay of Green Bay is the official name. So please join us at noon on September 5th. If you're here, please enjoy some refreshments. Thank you for joining us online, and I hope everyone has a great Friday.